welcome to this explainer. Right out of the gate today, we're jumping into something truly fascinating, a completely ground-up first principles framework that fundamentally reimagines how we understand and approach cancer. The source material we're unpacking here is a unique dissertation that actually sets aside traditional textbooks and established genetic dogma to look at this disease from absolute zero. It treats cancer not just as some random genetic accident, but as a terrain problem. So let's dive right in and see how this radically different perspective might just change everything we thought we knew.
To kick things off, we really have to ask a provocative question, one that challenges decades of established medical dogma. What if cancer cells aren't actually broken? The standard model we've all been taught tells us that cancer cells are defective. They're damaged, mutated beyond repair, right? But the author of our source makes a very different, very astute observation. Think about it. If these cells were truly damaged, they'd be weak. They would literally fall apart under stress, but they don't. Instead, they're incredibly tough, adaptable survivors. And honestly, that doesn't sound like damage. That sounds a whole lot like reprogramming.
And this leads us to a profound shift in perspective. There's this brilliant quote from the source material that states, "The cancer cell is not a traitor, it is a refugee." It posits that these cells aren't acting out of malice at all. They are simply trying to survive an environment that has completely abandoned them. See, in a healthy body, a cell trusts the system. It cooperates, it does its specialized job. But when the environment becomes entirely hostile, the cell basically decides the system is no longer protecting it. So what does it do? It jumps ship to survive.
Okay, let's move into Section 1, a flawless, desperate logic, or cancer as a terrain problem.
It's really important to notice the stark contrast here. Under the traditional standard model, the defective cell is the enemy. It's a traitor. But under this reversion theory, the cell is actually innocent, and its internal logic is flawless. It is responding perfectly to the corrupted, toxic signals it's receiving from the environment around it. The environment is the enemy here, not the cell itself.
So, to understand how this cell survives, we actually have to look way back into the past. The atavistic theory suggests that when conditions get really bad, cells just abandon multicellular cooperation altogether and revert back to ancient, independent survival programs. We're talking about programs that existed long before complex life, literally long before there was even oxygen in the Earth's atmosphere. They shift back to older, primitive forms of energy consumption, they evade the immune system just like ancient single-celled organisms would, and they prioritize their own immortality over cooperating with the rest of the body.
That brings us to Section 2. The Four Horsemen of Cancer, focusing on those environmental stressors.
There are four distinct universal forces, what the author ominously calls the Four Horsemen, that signal to a cell that its environment is just too dangerous for cooperation. First up is low oxygen, or hypoxia. Normally, oxygen signals that the environment can support complex life, right? When it drops, the cell panics and reverts to an ancient metabolism called glycolysis. Second is low pH. A highly acidic environment tells the cell it's essentially swimming in waste, so it protects itself by pumping even more acid out. Third, high toxins. Toxins corrupt cellular signals, forcing the cell to mutate just to adapt and stay alive. And finally, immune absence. When the immune system fails to enforce cooperation, these defecting cells survive and thrive. And the truly scary part, these four forces feed into each other, creating this massive, self-reinforcing cascade of terrain destruction.
So instead of viewing the cell as some evil or greedy invader, this framework suggests we view it as a desperate addict that has simply forgotten how to be healthy because of these corrupted signals. Low oxygen basically says, I can't trust my environment. Low pH says, I can't trust my community. High toxins say, I can't trust the system. It is just an addict trying to make it to the next day. And the only way to bring this cell back from the brink is to prove to it that the system is actually worth dying for again.
This leads us right into Section 3, the universal treatment backbone, all about restoring the environment.
If the environment is the problem, then logically, treating the environment has to be the solution. The author proposes a very specific three-step universal backbone to restore the body's physical terrain before even attempting any kind of immunotherapy. Why? Because immunotherapy often fails when the terrain is too hostile for immune cells to even function in the first place. This three-step process is designed to actively flip the environment from hostile back to healthy immediately upon diagnosis. Let's break down how each step targets the damage caused by those four horsemen. First, we target a protein called MCT4. Cancer cells use this protein like an exhaust pipe to pump acid out. By inhibiting it with a drug like AZD0095, you literally trap the acid inside the cancer cell, which kills it.
while the outside environment becomes more alkaline, which wakes the immune system back up. Then reoxygenation hits the cells with oxygen, putting severe stress on their ancient dysfunctional metabolism. Finally, molecular hydrogen comes in as a selective antioxidant. It protects the body's healthy T cells from the collateral damage of all that oxidative stress. It's a highly logical, completely synergistic triad.
Moving on to Section 4, testing the framework through clinical action and simulation.
So does it actually work? Well, in rigorous experimental simulations outlined in this source, this combined protocol showed extreme toxicity to aggressive cancer cells while leaving normal cells essentially untouched. They simulated this on MDA-MB231, which is an aggressive triple-negative breast cancer line, and the results? The combination therapy resulted in a massive 62-fold reduction in the concentration needed to inhibit the cancer cells compared to just reoxygenation alone. Acid was trapped inside the cancer cells, the outside environment successfully shifted to a healthy pH, and, this is the absolute kicker, normal, healthy cells, like the MCF10A line, were completely unaffected. That is the holy grail of cancer research right there. Selective toxicity based purely on the environment, not genetics.
Now, you might be wondering, what's the catch? When asked about the chances of this working all the way through human clinical translation to a durable cure, the author doesn't promise the moon. Instead, they offer an incredibly honest, objective estimate, a 10 to 30% chance of complete success.
Honestly, it is incredibly rare to see this level of transparency in medical research. It emphasizes that this is a testable blueprint, not some guaranteed miracle cure or snake oil. This is a falsifiable protocol relying on strict logic. If the tests fail at any of the predefined gates, the hypothesis is disproven. Simple as that. But that 10 to 30% probability, that is a deeply meaningful number for a disease that has historically defied so many standard treatments. It's an approach that absolutely demands to be tested.
Let's move to our final section, Section 5, the Master Plumber's Blueprint, a testament to first principles.
Prepare to be surprised here. The most astonishing fact about this highly structured, rigorous medical dissertation is who wrote it. You might expect a team of Ivy League geneticists, but actually, scratch that. It was authored by Nicholas D. Coleman, a master plumber from Texas, relying purely on independent logic and observation. He has no institutional funding, no multi-million-dollar lab, and no medical degree. He simply used first principles reasoning, breaking a highly complex problem down to its most basic foundational truths to identify patterns that the established paradigm was just completely missing.
His unique background in building and fixing physical pressure systems gave him the ultimate first principles lens to view the systems of the human body. He builds pressure for a living. He implicitly understands flow, toxicity, and environmental systems on a practical, physical level. As he states in the text, he just observed the patterns, asked the right questions, and refused to accept the dominant narrative when it simply didn't explain the reality of what these cells were actually doing.
As we wrap up this explainer today, I want to leave you with one final, provocative thought to chew on. If the environment dictates the behavior of our internal cells to this degree, how must we change our own external environments to truly heal? This framework challenges every one of us to stop looking at cancer as just a random, inescapable genetic lottery and start looking hard at the terrain we are creating inside our bodies. From the oxygen we breathe, to the toxins we absorb, to the chronic stressors we endure every single day, the terrain is everything. The logic is sound, the blueprint is laid out, and the path forward is clear. So maybe it's time we start fixing the environment.